In this session, we are going to see about chemistry of amino acids. So these are the objectives for this session. You should be able to classify and list the amino acids present in proteins, explain the biomedical importance of those amino acids, describe the structural organization of protein, just an overview, okay, and then describe the importance of something called denaturation of proteins. Okay, fine. So what are amino acids? Amino acids are building blocks of proteins, and then each amino acid is genetically encoded. Okay, that is coded by a gene. One codon will code for an amino acid. For example, AUG is a codon which codes for methionine. Okay, a set of 20 amino acids constitute almost all proteins in our body. Okay, an amino acid, as name suggests, contains an amino group and a carboxyl group. Okay, so these are the 20 amino acids which makes up most of the proteins in our body. Okay, leucine, lysine, isoleucine, valine, methionine, phenylalanine, threonine, tryptophan, arginine, histidine, glycine, alanine, serine, cysteine, asparagine, glutamine, aspartic acid, glutamic acid, tyrosine, and proline. And these 20 amino acids are also called as standard amino acids or alpha amino acids. Okay, fine. So this is a general structure of an amino acid. Amino acids are made up of an amino group and the carboxyl group. Okay, and there's one R group and there's one hydrogen group. Okay, and this R is nothing but a side chain. Okay, and amino acids differ from one another based upon their side chain, which is designated as R here. Okay, that is, the side chain will determine the charge of that amino acid as well as the polarity of that amino acid. So the nature of the side chain is very important for an amino acid. Okay, all amino acids found in proteins are called as alpha amino acids because both the amino group and the carboxyl groups are attached to the same carbon atom. Okay, almost all amino acids are called as alpha amino acids except proline. I will tell you why. Okay, fine. So each amino acids are represented by symbols using letters. It can be either a 1-letter symbol or it can be a 3-letter symbol. For example, tyrosine is represented by Y, capital Y, or it can be written as TYR. Alanine can be written as A or ALA. Histidine as H or HIS. Arginine as R or ARG. Okay, so this is how amino acids are represented. This is the system of representation of amino acids, either a 1-letter symbol or a 3-letter symbol. Okay, so 20 amino acids make up all proteins in the body, and these amino acids are classified based upon the nature of the side chain, either based on the chemical structure itself or based on the charge as charged and non-charged amino acids, or based on the polarity as polar amino acids and non-polar amino acids, then based upon the nutritional importance. Okay, and based upon the metabolic fate of the amino acid when they break down in the cell. Okay, this is how amino acids can be classified. Okay, nature of the side chain, nutritional importance, and metabolic fate. Okay, for example, based on the chemical structure. So first is aliphatic amino acids. They are nothing but simple branched or branch chain amino acids. For example, valine. There is a amino group, there is a carboxyl group, and this is a simple R chain. Okay, this is valine. Aromatic amino acids. If the R group, that is side chain, has an aromatic ring in its structure, for example, tyrosine is an aromatic amino acid. There is something called amino acids, which are proline. Okay, where if you remember correctly, I said almost all amino acids are alpha amino acids because amino group and the carboxyl group are attached to the same carbon. Here, see here, there is a carbon, alpha carbon is here, it's attached to OH group, and this alpha carbon is attached to NH group, which is part of a ring structure. Okay, here the amino group is not separate like this, like what you see here. This is called amino acid. Property of this makes proline to be present in the bends of alpha helices because it cannot be involved in bond formation. Okay, that's why in the bends of the alpha helices, okay, you will see proline. If you see proline in a chain, there will be a kink in that area because of this property. Okay, so almost all amino acids are alpha, whereas proline is said to be amino acid. Okay, because of this structure. Okay, this group is called secondary amine group. Okay, fine. So that's about the chemical structure. Okay, now based on the charge they carry. okay, as acidic amino acids. Okay, for example, this is aspartic acid. Basic, this is histidine. Okay, neutral, glycine. So aspartic acid will have a acidic, that is negative charge. Basic amino acids will have a positive charge, and neutral amino acids will have none of the charges. Okay, that is how they are classified based on the charge they carry. Okay, then based on the polarity. Okay, it can be either non-polar or polar. Okay, so non-polar, the best example is alanine. Okay, and the polar, it can be either cysteine or it can be lysine. Okay, so non-polar are hydrophobic in nature, polar are hydrophilic in nature. This is how they are classified based upon the polarity. Okay, next, based upon the nutritional importance. Okay, out of 20 amino acids I have listed out, only 12 amino acids can be synthesized our own cells in the body, and there are some amino acids which cannot be synthesized in our body. Okay, and those amino acids should be supplied only from the diet. Okay, and those amino acids are called as essential amino acids. The amino acids that can be synthesized in the body are called as non-essential amino acids. Those which cannot be synthesized, which has to be supplied from the diet, are called as essential amino acids. Okay, these are those essential amino acids: leucine, lysine, isoleucine, valine, methionine, phenylalanine, threonine, and tryptophan. Okay, these 8 amino acids should be supplied only from the diet. Okay, they are called as essential amino acids. There are something called semi-essential amino acids or conditionally essential amino acids. There are some amino acids that are synthesized in the body in small quantities only during certain conditions. For example, say pregnancy, lactation, and childhood. The body will try to do this only during those conditions. That's why they call it semi-essential. Examples are arginine and histidine. Arginine can be produced during urea cycle. Okay, so that's the reason for semi-essential amino acids. Okay, then the fourth way of classifying an amino acid is based upon its metabolic fate. Okay, what is it? See, when amino acids break down, it has an amino group and a carbon skeleton. Okay, the amino group will be converted to form ammonia, which will be detoxified to form urea. That's different. Okay, and this carbon skeleton will be broken down to form intermediates. Okay, and those intermediates will determine the metabolic fate of an amino acid. For example, if the intermediates that is produced during this carbon skeleton breakdown produce only glucose, then they are called as glucogenic amino acids. Examples: isoleucine, tyrosine, tryptophan, and phenylalanine. They are purely glucogenic amino acids. Whereas some amino acids can break down to form only ketone bodies. They are called as ketogenic amino acids. There are only 2 amino acids like that, leucine and lysine. Okay, that is, to put it in clear words, amino acids, when they break down, they can form intermediates, and those intermediates can be used either to form glucose or ketone bodies. If they form glucose, then they are called glucogenic amino acids. If they form ketone, they are called as ketogenic amino acids. These are the examples. So only 6 amino acids are covered. What happened to the remaining? The remaining amino acids can produce either glucose or ketones. That's why they are called as glucogenic and ketogenic amino acids. For example, serine, valine, glycine, and alanine. So they can produce either glucose or ketone or both. Okay, so that's why they are called as glucogenic, ketogenic, or both glucogenic and ketogenic amino acids. Okay, fine. So there are certain properties of amino acids which we should know. Ionic forms of amino acids. Okay, in acidic solutions, amino acids are cationic. That is, they carry net positive charge. Okay, whereas in basic solutions, amino acids are anionic. That is, they carry net negative charge. Okay, fine. So what is isoelectric pH? Isoelectric pH is a pH at which amino acids will carry no net charge. That means they'll have equal number of positive ions and equal number of negative ions. So they will be electrically neutral. Okay, the pH at which the amino acid exists like this is called as isoelectric pH. Okay, and the amino acid that exists at this state is called as Witter ions. Okay, why this is important? This isoelectric pH is employed in precipitating the proteins out of a solution. Okay, so proteins can be separated when you bring that protein towards isoelectric pH, so they cannot move on either side, they will settle down. When you isolate that protein and you read about that protein, you can do some research on those proteins. Okay, so that is what is important when it comes to isoelectric pH. Okay, fine. Next, what are the functions of amino acids? See, amino acids are linked together by peptide linkage to form proteins. Okay, amino acids as such can give rise to various products. For example, tryptophan can give rise to serotonin. Histidine can give rise to histamine. Both are neurotransmitters. Tyrosine can give rise to thyroxine and catecholamines. Both of them are hormones. Okay, then glycine is part of heme synthesis. So glycine can also be, is one of the important products derived from glycine. Okay, So when the amino acids are broken down, that is when the proteins are broken down, they will yield almost 5 to 10% of the total energy required by the body. Okay, so these are the functions of amino acids. Okay, fine. Now, there's a little bit overview on the structural organization of proteins. Okay, the simple structure is primary, secondary, tertiary, and quaternary. Okay, what are primary? Primary structure is a sequence of amino acids in a protein. Okay, just a sequence of amino acid 1, 2, 3, 4, 5, 6, 7, 8. It's a linear sequence. It's a chain, straight chain. Okay, and they are joined together by peptide bond. That is primary sequence, primary structure. Okay, see here, primary, this peptide linkage, the peptide bond is a double bond like character. They are rigid and planar. Okay, that's a characteristic of this peptide bond. Okay, fine. What is secondary structure? Secondary structures, amino acids are linked together to form a polypeptide. And this polypeptide chain will assume a helical conformation or a sheet conformation. They will not assume a random three-dimensional structure. Okay, it undergoes a regular arrangement to establish a steric relationship between the amino acids. Okay, and this arrangement can be either a helical form, which is also called as alpha helix, or it can be a beta sheet, which can be called as a beta sheet form. Okay, collagen, an excellent example of a protein which consists of alpha helices. It's a triple helical molecule. Okay, fine. See here, this is a structure of secondary structure for protein. Secondary structure of protein is stabilized by this bond called intra-chain hydrogen bonding. They can ask you which bond stabilizes secondary structure, intra-chain hydrogen bonding. Which bond stabilizes primary structure, peptide bond. Okay, fine. That's about secondary structure. This is one of the secondary structure, alpha helix. There are many structures like beta sheets, loops, turns, beta bends. Okay, so that's about the secondary. What is tertiary? Tertiary is a single polypeptide chain undergoes folding. It will fold among itself to form a three-dimensional structure. Okay, they are also called as domains. Okay, and this protein folding, which happens in the endoplasmic reticulum, Golgi bodies, etc., is facilitated by disulfide bonds and hydrophobic interactions. Okay, so that is about tertiary structure. For example, myoglobin, albumin. Myoglobin is made up of only one globin chain, which folds upon itself to form a globular structure. Okay, which is stabilized by protein folding and stabilized by disulfide bonds. Okay, that's it. So the bond which stabilizes the tertiary structure is disulfide bond. Okay, fine. Quaternary structure. The quaternary structure, two or more polypeptide chains will come together and they're held together by non-covalent bonds to form a complete functional protein. The best example is hemoglobin. Hemoglobin is made up of 2 alpha chains, 2 beta chains. It's made up of 4 globin chains. 4 globin chains will come together. They will be held together by non-covalent interactions. Okay, that is called as quaternary structure. Best example is hemoglobin. Okay, so this is a summary of structural organization of protein. Okay, we have a primary structure, which is a linear sequence of amino acids joined together by peptide bond. Then we have a secondary structure in the form of alpha helix, stabilized by hydrogen bonds. Then we have a tertiary structure. A single polypeptide chain will fold upon itself to form like this. Then there is a tertiary structure which is made up of more than one polypeptide chain. For example, 4 polypeptide chains in hemoglobin. Okay, so this is about quaternary structure. Okay, so this is how proteins can be classified based upon their functions. For example, it can be a structural protein, it can be a catalytic protein, it can be a contractile protein, transport protein, regulatory protein, or storage protein. Okay, or based upon its composition, it can be a simple, conjugated, or derived protein. Based upon the size and shape as fibrous protein or globular protein. Nutritional value, nutritionally rich protein, which contains almost all the 20 amino acids in it. Or nutritionally poor, it contains only few amino acids in it, not the major part of amino acids, which is essential for protein synthesis in our body. This is how you can classify protein. Okay, what are the functions of proteins? Functions of proteins: defense proteins. Antibodies acts as the defense against the microorganisms. Structural protein: collagen, elastin, keratin. Catalytic proteins, example enzymes. All enzymes are proteins except few. For example, ribozymes, they are not enzymes. They are made up of RNA. Okay. Transport proteins: albumin, transferrin. Albumin transports drugs. Bilirubin. Transferrin transports iron. Okay. Contractile proteins, example actin and myosin. Storage proteins, example ferritin. It stores iron. Buffers, example plasma proteins. Albumin acts as a buffer. Hemoglobin acts as a buffer. Okay. Then regulatory proteins, example hormones, insulin, growth hormones, they're nothing but proteins. They have regulated the various metabolic pathways in your body. That's why they call as regulatory proteins. Then conjugated proteins, for example, lipoproteins, which carry triglycerol and cholesterol. Then glycoproteins, which acts as the blood group antigens, which acts as the receptors in the cell membrane. Okay, these are the other functions of protein. Okay. Now, there's one process called denaturation. What is denaturation? Denaturation is a process where the protein will lose its secondary structure and tertiary structure and quaternary structure. Only the primary structure will be intact. And this process of unfolding and disorganization of the protein secondary and tertiary and quaternary structure is called denaturation. Okay, denaturation is caused when you subject that protein to heating, or when you treat that protein with strong acids, or mechanical mixing, or detergents. Okay, denaturation is usually an irreversible process. When you denature a protein, it is irreversible. That's it. All the structures will be lost, only the primary structure will be intact. Okay, for example, you see this egg albumin. You boil it, it will become like this. Or you put it on a fry pan, it will become like this. When you cool it or when you remove heating, it is not going to come back to its original position. This is one example that denaturation is not always a reversible process. Whereas, if you heat a milk, it boils. It still remains liquid only. Okay, why I am telling you this? Because milk is made up of casein, which is a milk protein. Where you are heating a protein, it is undergoing denaturation, but if you cool it, it is coming back to its original form. Okay, so that's why it is not always a reversible process. For some proteins, it is reversible. For some proteins, it's not reversible. That's it. Okay, fine. What is the importance of denaturation? Protein on denaturation will lose its biological property. Why? The biological activity for protein depends upon its quaternary structure and tertiary structure and secondary structure. If those structures are lost, proteins will be no longer functional. So, denatured protein is no longer functional. For example, some proteins such as insulin, okay, you would have seen patients with diabetes taking insulin injections. Okay, we always advise them to keep those insulin injections in the fridge. Why? Because when you keep it outside, because of the temperature, the insulin can go for degradation or denaturation. So, insulin will lose its activity. Even if you take that insulin shot, it is not going to decrease your blood glucose level. Okay, that's the importance of maintaining proteins at correct temperatures. Okay, because the extremes of temperature can denature the protein and make them lose its biological activity. Okay, that's one important point. Okay, enzymes which catalyze the biochemical reactions lose their functions at high temperatures. Okay, a simple example is this. Okay, for example, you take gastric enzyme called lipase. Okay, lipase is active only in acidic pH because stomach environment is extremely acidic. If you take that lipase and put it in the duodenum, it is not going to act because duodenal pH is alkaline. So they are because at that pH they can get denatured. Okay, so that's one point. Okay, fine. So to summarize, a set of 20 amino acids act as building blocks of proteins. Okay, amino acids can be classified based upon the nature of their side chains and their initial requirements. For example, based on the side chain as charged, uncharged, polar, non-polar. Okay, aromatic, aliphatic, and amino acids. Okay, based upon the nutritional importance as essential amino acids and non-essential amino acids. Okay, and based upon their metabolic fate as glucogenic or glucogenic and ketogenic amino acids. And proteins are organized at 4 levels: primary, secondary, tertiary, quaternary. Primary is for peptide linkage, secondary is by hydrogen bonding, tertiary is by disulfide bond, quaternary is by non-covalent interactions. Proteins perform various functions in your body. Okay. At last, we saw about denaturation. Denaturation is a process by which protein will lose its secondary structure, tertiary structure, and quaternary structure. Okay. Only the primary structure will be intact. Okay. And denaturation is not always a reversible process. When a protein gets denatured, the functional activity of that protein will be lost. Okay. That's it. With this, I'll close this session. Thank you.